To take action towards a better section on technology can provoke a rapid introduction to a better quality not only of living, it can bring better circumstances to our current standards. Genetic engineering can be useful in an indefinite number of ways such as applications, including scientific research, agriculture, and technology. Advancement in medicine is a critical point that can determine a civilization's stability and termination of viruses. Most importantly, genetic engineering, when it's based with moral responsibility, can sustain the humanity's interests towards a better path. Gaining access to this type of technology can also strengthen the proteins that help an individual's digestion, organ diseases, or produce a vaccine cure. This film will address what the GMO's processes help improvements to be brought to current civilization and the type of diversity in genetics encountered. The future of GMOs is distinguished by subjection of genes, modifying a constant loyalty and beneficial improvements to the humanity path. The constraint of making corms a unique primary state in order to make bigger amounts of this starts with the fact that GM plants risk to hammer in unpredictable ways. Some of these vegetable crops produce their pesticides from insects, which can be non-harmful to humans, contrary to chocolate to dogs, which seems to damage their interior. But actually, plants that allow their own nitrogen allow nutrients to absorb more carbon dioxide, making this reliable to climate conditions, and finally, be able to reverse climate change, making imagination the last option for most effective crop. GMO could be our last integrate to biosphere salvation.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
The improvements can be done by editing DNA before even being born, although this can differ in a big amount, bringing with them different characteristics. By creating the right genes, good iomes will be created that live and are used, such as biosensors, explosive detectors, and run drug deliveries in a human body. In the first place comes medicine, which can make the production of these in greater amount and more efficient, or even prevent for starting. Genetic engineering can bring modifications from a simple embryo editing to a transformation into a gamete, meaning this that bringing a fluorescent color, changing the hair color, or giving a different eye color is not the only option.